Hi everyone, thanks for coming. So, I'll talk a bit about an in vitro hydrogel scaffold co-culture of lymphangioliomyomatosis, or LAM. LAM is a fatal lung disease for which there's currently no cure. It's predominant in childbearing age women and is characterized by the invasion of mutant smooth muscle cells into the lung area, which can lead to lung puncture, collapse, and then a need for a lung transplant. So, in the interest of finding a cure, there's been a push toward high throughput screening for a drug candidate. However, in order to achieve this, it requires accurate in vitro modeling of lamb cell behavior, which then entails replication of the natural extracellular matrix or ECM environment so we can get the cells to behave as they would in the body. The current standard, however, for high throughput screening is 2D plates, such as transwells, which fail to really capture the complexity of the local lung microenvironment. And because of this, there's been a move towards three-dimensional hydrogels being water-swelled polymer networks, which can be mechanically and chemically tuned to be similar to tissue. But in order for these systems to be viable, they must be able to support three critical aspects of cellular behavior, namely cell viability, cell migration and invasion, as well as cell-cell interactions and signaling. And with this, our goal was then to create an in vitro three-dimensional hydrogel tissue model for LAM drug screening. Let's move down to figure 1 here. So our lab created a hydrogel based on two biopolymers and polypeptides. So our two biopolymers, hyaluronic acid, or HA, which is biologically relevant because of its presence or overproduction in the lamb and tumor environments, and methacellulose, or MC, which is beneficial because of its ability to form physical crosslinks through hydrophobic junctions, which I'll talk about in a bit. These are complemented by the two peptides, the one here in green on the bottom, which helps the cells adhere to the matrix, and the ones on red, which is degradable by enzymes secreted from the lamb cells, which will help the lamb cells invade, and it also cross-links the biopolymer components together. So in figure 2 here, this components allows us to create a dual chemically and physically cross-linked gel material. Firstly, through chemical cross-linking gives us mechanical stiffness to the material, which not only makes the gel self-standing, but also helps the cells identify and adhere to the substrate. And physical cross-linking down here, as I mentioned earlier, is mediated through the MC component and imparts viscoelastic properties on the material, which, as previous studies have shown, make the material more conducive to cellular migration and invasion. And down here in figure 3, so we tested this in a 384 well plate format where we would have the gel inside the wells and seed proliferally lamb cells on top and culture them over 5 days. Over this period, we noted that the lamb cells remained adherent and viable for the entire period. And moreover, by 5 days, we stained and imaged them and found that they had invaded considerably into the middle of the gel matrix, suggesting that of the original three criteria, we had the current gel formulation can support viability, adhesion, migration, and invasion. However, being a monoculture, this system still does not yet demonstrate the ability to model cell-cell interactions. And we think this is important for LAM because LAM exists in the lung where there are multiple other cell types, such as other stromal and immune cells, which could affect the LAM behavior. A recent study by a different group found that macrophages actually represent the largest proportion of the cell population in late-stage LAM cysts. And because of that, we were interested in upgrading our gel to a co-culture model of both LAM cells and macrophages together in the single material, specifically where we would culture or encapsulate macrophages in the bulk of the gel, have LAM cells on top, and allow them to invade downwards upon they would contact each other. And we would then analyze the cell-cell contact and its effects on the cytokine profile of both cells, as well as their phenotype based on flow cytometry. However, we found that several adaptations needed to be made to the original gel in order to make it amenable for such a kind of a co-culture. Because we originally designed the gel to be tailored to lamb cells, there are a few improvements we need in order to make macrophages happy as well. Specifically, we need to redesign the adhesive peptides since the original sequence of vitronectin is specific or designed for LAM. And also we found that macrophages are liable to fall out of the current gel because the porosity is large and it was created for the relatively large LAM cells, but macrophages are smaller and so would like a much smaller mesh size. And because of that, we're looking at tuning the physical and chemical cross-linking proportions to make it more viscoelastic, which we think will solve the problem. So in conclusion, with the original goal of creating a three-dimensional hydrogel tissue model for LAM drug screening, we created an HA and MC hybrid material with a dual cross-linking mode of both chemical and physical, which allowed us to incorporate a viscoelastic cell invasion promoting component, and found that this allowed LAM cells to be adherent, viable, and also able to invade their matrix. And our current work involves upgrading the gel chemistry to support a co-culture with macrophages and LAM cells. Apart from LAM specifically, our work also has important implications on the disease modeling field in general. Namely, we're one of the few gels that contains both a physical and chemical cross-linking component, and so is therefore highlights the importance of considering the viscoelastic component when trying to get cells to invade. And also, for once we get the co-culture system to work, it will highlight it as a valuable strategy of considering cell-cell interactions when trying to create a viable cell disease modeling platform. So I'd like to acknowledge funding from NSERC as well as everyone from the Shortkit Lab for helping make this project possible, and I'll be happy to answer any questions.
Thank you, Spencer, for your presentation. Judges, if you have any questions, feel free to unmute yourselves.
Yeah, here is Charles. And Spencer, very cool project. I wonder, you mentioned that the LAM system may be applicable in the future for disease modeling. I wonder that how representative that model will be, thinking about people, I'd say young people, old people, right? I think, can you comment on that?
Yeah, so we think that by incorporating these three factors, that the gel is already pretty advanced in being able to replicate what LAM cells would normally experience in their environments by incorporating their ability to move as well as their ability to sense with or interact with other cells. Although we do think there are maybe a few limitations to this. Sorry, a few limitations to this network. In that, for example, LAM cells naturally form cysts, that kind of ball structures, and not just like freely dispersed, kind of like in a monolithic kind of gel. So that may be one limitation to accurately modeling what happens in the disease. But we think that compared to the other kind of standard models out there, our model is pretty well advanced in mimicking what LAM cells would normally act in the disease state.
Very good. Thank you.
Hey, Spencer, I have a question. This was more on the research process. So I was wondering, what are the implications or what are the next steps for this research to be implemented in clinical studies? So what would the process be like moving forward?
Yes, so we haven't thought yet much about moving it to clinical studies. We were more thinking about getting the co-culture system to work. So as I mentioned with the future directions, we will be tuning the chemistry of the chemistry of gels to help it become more broadly applicable to different kinds of cells. That's be our main kind of next steps. But once we get that done, we hope to demonstrate its ease of use for high throughput screening, for its ability to say seed it on a 384 well plate and be able to rapidly screen different drugs on it. And yeah, so that would be our main next application once we get everything working.
Thank you.